Hello and welcome to BioRules video lessons higher level edition. So this here is going to be a high level class, but the standard levels are also going to be learning this so they can say I know high level stuff as well. Okay. Today we are going to be taking a look more closely on photosynthesis. If you watched the very basic photosynthesis overview, you have an idea on how photosynthesis happens, but let's take a look on everything together here in a high level view. For us to talk about photosynthesis, we have to talk about chloroplasts. Just a quick overview here on the chloroplast. Later we are going to take a look more in deep on the structure of chloroplast and how it helps it to perform its function. Okay, so chloroplast, it has two membranes. So endosymbiotic theory, it was engulfed by another cell, kept the two membranes, it has its own DNA, circular naked, it has its own ribosome 70S, so it is basically a bacteria living inside of plant cells. It also has a system of membranes called thylakoids. Those little coins over there, they are roundish bags of membrane that has making the chloroplast to have a very big surface area of thylakoids, but a very small lumen inside of those thylakoids. So here now we are going to be having the structures, the outer membrane, inner membrane. Okay, the cytoplasm of the chloroplast is called stroma, and you have there the thylakoid. The thylakoid has a lumen. So the lumen actually is a very narrow space, is equivalent to the mitochondria's intermembrane space. The intermembrane space is a very narrow space where the plants accumulate, mitochondria, where the mitochondria accumulates protons, in the chemosmosis and all the stuff. Here, the thylakoid will do the same. The thylakoid lumen is going to be a very narrow space with a huge surface area full of proteins to be able to do photosynthesis and accumulate protons inside of the lumen to make things to happen. Okay, a bunch of thylakoids together is called a granum. They are connected by lamella, so they are all interconnected together. Okay, and as I told you before, we have circular naked DNA and we have 70S ribosome. This is the structure of chloroplast, because now that we're going to see the light-dependent reaction, the light-independent reaction, we need to talk about thylakoid, stroma, and the lumen. Okay, so another view over here for people that like a prettier drawing than mine. But now let's talk about cell respiration. Oh wait, I thought we were talking about photosynthesis. Yes, for us to talk about photosynthesis, let's make a quick remind of cell respiration. Okay, so we have there the inner membrane, the matrix, and the intermembrane space. NADH, which is bringing electrons and protons from the glycolysis link reaction and Krebs cycle, electrons and protons that have been stolen from chopping down glucose into carbon dioxides. Now they are brought into here and they give away the hydrogen. Remember, oil rig, oxidation is loss, reduction is gain. So NADH got oxidized, gave its electron, lost its electron, so became NAD+. So in that process, electrons go around and they move around these proteins, and the movement of these electrons is enough to pump protons into this pump, which is an active transport happening, that instead of being moved by ATP is being moved by electrons. The electrons keep carrying on, okay, or they are also going to be releasing electrons, and these electrons pump more proton, and then they go to another protein, and there they pump more proton, and then the electrons have to go somewhere. In cell respiration, electrons go with oxygen and protons, and they end up forming water. Okay, so this here is what happens to electrons into cell respiration. Then, of course, lots of protons get accumulated, protons. We have a gradient of protons into this tiny little space called intermembrane space that will make a gradient and will make the protons to move down, creating kinetic energy of the ATP synthase, which is capable of transforming this kinetic energy into ATP, gluing ADP plus P, making ATP. So what we had here was that the electrons energy over here that were given to the membrane and end up in making water, they are used to make ATP. And then this ATP is what you use for cell respiration. Okay, so this is the mitochondria's electron transport chain, and from now on we are going to be talking about mitochondria. Okay, so the process that light-dependent reaction is going to happen is called photophosphorylation. Photophosphorylation because we phosphorylate, we put phosphate into the ADP, making ATP or ATP formation, and we are going to be doing that with light. So how that happens? If you remember cell respiration that I just showed you, the high level of cell respiration, what happens there in the mitochondria, the photosynthesis is going to be pretty simple, okay? We have here the membrane of the thylakoid, which is the equivalent of the cristae of the mitochondria, the equivalent of the inner membrane that contains a series of proteins, including a photosystem 2 and a photosystem 1. Those are proteins that are going to be making photosynthesis to happen, okay? And we also have ATP synthase there on the right. So this should be quite familiar for you, okay? On the top, thylakoid lumen, so the tiny little space in between the thylakoid membrane. So you have the thylakoid membrane, you have the stroma, which is the equivalent of the matrix. So how things are going to happen in here? First thing, light hits photosystem 2, which contain chlorophyll. Chlorophyll is excited by light, electrons jump out of it, so it suffers oxidation, the loss of electrons, and by doing that, they force water to also suffer oxidation, giving electrons to the chlorophyll, so replacing the electron that was lost by light, and releasing oxygen as a byproduct. So the oxygen produced by photosynthesis, that's vital for many living organisms on planet Earth, and that change planet Earth when photosynthesis starts happening in a large scale. This oxygen is produced at this very moment, the moment of breaking water with light, also called hydrolysis. Hydrolysis, hydro, water, lysis, breaking water. Okay? In this process, some protons are produced and they start accumulating there in thylakoid lumen. The electrons that have been released, they move now from the photosystem 2 to another protein in the thylakoid membrane, and in that protein, it energizes that protein, that pump, to pump protons in the thylakoid lumen. So this here should sound familiar because it's equivalent of what happens in the mitochondria, electrons moving, pumping protons. These electrons now, they are going to be less energetic. They gave some energy in order to be able to do this pumping. So these electrons have to be re-excited. So that will happen here in the photosystem 1. Light will re-excite chlorophyll that's present in there, that will re-excite these electrons and put it to move now very excited. And these electrons that are very excited now, they can be accepted by an electron acceptor, which is the NAD, or in this case the NAD of photosynthesis, NADP plus, becomes NADPH plus H plus. So basically we just reduced NADP to make NADP to receive electrons and protons, and now we have produced these guys over here. We have highly excited electrons that can be used in the near future. This here keeps happening and happening and happening, and goes accumulating protons in the thylakoid lumen, and accumulation of protons in a narrow space creates a gradient. These protons are desperate to move away. They diffuse out of that if they are let. They diffuse through ATP synthase, and ATP synthase uses the kinetic energy of itself twisting around to phosphorylate ADP plus P, making ATP. So we just finished this mechanism here in the thylakoid lumen, where now we have NADPH, or highly excited electrons, and also protons, and we have ATP. Now we have energy to carry on the remaining of photosynthesis. Okay? If you want to see this in another diagram, we have here photosystem 2, light excited in there, being used to break water, hydrolysis, producing oxygen that goes away, producing also protons. These electrons, they move around and they help to pump protons more in the thylakoid membrane. The electrons are re-excited, so they end up there making NADPH plus H plus, and we end up having here a bunch of electrons. These electrons move down through the ATP synthase producing ATP. So what we have loss of NADPH and we have loss of ATP. This is the light-dependent reaction. It's using light to break water and to produce NADPH and to produce ATP. This is what we have. So if you put this in the photosynthesis formula, it will look like this. Light is used specifically to break water into oxygen and also releasing electrons and hydrogen protons. That will be taken by NADP, that becomes NADPH, and also in this process, the ATP synthase uses the extra protons that have been accumulated over there to produce ATP. So we have now ATP being produced and NADPH. So we have two highly energetic chemicals that have been produced from breaking the water, making oxygen with the use of sunlight over here. This is photophosphorylation. It is quite similar in some ways to cell respiration, but it produces ATP in the same fashion as cell respiration. It's the movement of protons down a gradient through ATP synthase produce that. But it's the opposite in terms of electrons. Instead of using NADH to give electrons to be able to pump protons, the breakdown of water makes protons and the movement of electrons pump protons and now re-excite the electrons to make NADPH, and these two guys are going to be used in the end. So photosynthesis in this stage is half the same, half the opposite of cell respiration. Okay? I hope you got the idea. We are going to have class so we can discuss all this, bring your doubts, bring your discussion. Let's make sure that you get all this very high level mechanism. Okay? Now do the quiz, see if you got the idea. If necessary, you watch the video again, pausing and thinking, making your notes so you can make a good quiz. Okay? See you in class. Bye-bye.